Hi everyone, my name is Natalie, and I'm a board member with Propel a Cure for Crohn's disease. I'm also a patient with Crohn's disease. I've struggled with the symptoms of IBD for 27 years. 27 years is a long time, and it's really disheartening to see people continue to be diagnosed with Crohn's today, and we still don't understand what the cause is or what's contributing to the condition. Our community desperately needs new treatments. We desperately need prevention, and we desperately need a pathway to a cure. And the only way we're going to get there is with investing in sound research. That's why I'm so proud to volunteer with and support Propel a Cure. This past fall, we've been working on a really exciting project that I'm thrilled to share with you. We've been interviewing researchers who are doing groundbreaking, novel research in the Crohn's disease space. This research will deepen our understanding about what's causing the condition and potentially pave the way to new treatments, prevention, and cures. Our first interview is with Dr. Brian Coombes, a Canadian researcher who's studying adhesive invasive E. coli, a pathogen that's associated with Crohn's disease. It's this exact type of research that Propel a Cure wants to support, but we can't do it without your help.
Well, thank you again for joining us, Dr. Coombes. It's really a pleasure for us all to be here speaking to you and to get to pick your brain a little bit about Crohn's disease and your research. So we'll jump right in and hopefully just have a conversation about what you're doing and sort of where you see the direction of the research leading. But really, I just want to start by asking you to tell us a little bit about your research, where it's headed, and then, what your hopes are sort of for the future of your research in Crohn's.
Sure. Thanks very much for having me. It's a pleasure to be here. So our research in IBD we've been attacking this problem quite decidedly from a microbiological point of view. I'm a microbiologist and a biochemist by training. I did my PhD in biochemistry and medical sciences and a postdoctoral fellowship in bacterial pathogenesis. So I'm very interested in how bacteria evade our immune system, how they make us sick, how they make us healthy even as well, which is really a newer kind of concept that the microbes in and on us are actually beneficial to our health. So when we first started working on, I got thinking about actually working on IBD in the lab. Gosh, it was probably 2008, 2009, I guess, a long time ago. And we got a small seed grant from Crohn's and Colitis Canada to start to look at these very unusual bacteria that a French group had been defining as associated with Crohn's disease. We call them adherent invasive E. coli, or AIEC. And back then, there wasn't much known about AIEC and almost no research going on in adherent invasive E. coli research, and very little in Canada, and also very little in the United States actually as well. So we thought it was kind of a, kind of a good niche for us to sort of slide in and apply some of our microbiology background to as a really serious problem. So that's where it all started. And we got very interested in these adherent invasive E. coli once we got going, because quite quickly we realized that they're very interesting and unusual bacteria. And they're highly associated with Crohn's disease. They're about 6 to 10 times more likely to be found in Crohn's patients than healthy individuals. The interesting thing as well is that they're not missing from healthy individuals either. So although they're more prevalent in folks with IBD, healthy individuals also can harbor AIEC in their GI tract, and it's a much less prevalent. Only about 6% of people have AIEC in their GI tract. These are healthy individuals. And so we're very interested in to understand how these bacteria go bad, and why are they more associated with Crohn's patients than healthy individuals? And what is it about the Crohn's environment in the gastrointestinal tract that allows these particular kinds of bacteria to proliferate and expand and be disease associated. I'm hesitant to say they're causally related at this point, just because I don't think the evidence is there, but certainly it is unequivocal that the association is very strong, and it is significant. And the association has been borne out in epidemiological studies essentially worldwide now. They were these bacteria were first discovered and isolated by the lab of Arlette de Formichaud in France, and since then have been reproduced, in many countries of the world where people are studying Crohn's disease. So the association, I would say, is unequivocal, and it's very sound. Now our minds are sort of turning to, are these bacteria taking advantage of an environment in the gut that is conducive to their growth? Are they causing the inflammation itself, or are they perpetuating it once it gets started? So there's a lot of questions, there's a lot to learn, but we've actually come a really long way since 2008, I guess, is when we started this work. Lots of things the lab's been really productive trying to understand these questions and understand the infection biology associated with AIC.
So that's fantastic. As an introduction, I think you actually answered a lot of my questions just in that one introduction. But I guess my next question is then, so where do you go from here? What's next in terms of what you are looking at and what you would ideally like to be doing to answer some of these difficult questions?
Yeah, I mean, I touched on the notion that I'm hesitant to say AIC bacteria are causal in Crohn's disease, but certainly the association is there. And so, as you well understand, and your listeners will well understand, Crohn's disease is very enigmatic in the sense that, predicting and understanding who is at risk for Crohn's disease and who might ultimately go on to develop Crohn's disease is very, very hard to predict. Familial history only goes so far to explain some of the risk. And although, first degree relatives are more likely to be at more risk for Crohn's disease, it certainly doesn't explain it all. And even monozygotic twins, who you'd think if one has the disease, the other should, if it was an entirely genetic disease, that's all obviously doesn't happen either. It only is prevalent in both twins, about 25 to 45% of the time. So clearly there's more going on than just genetics, although genetics does play a role. And I've sat in a lot of IBD conferences, and many people, as their first slide, will put up this sort of Venn diagram where you've got microbes, environmental factors, and genetics all sort of coming together, and in the middle you've got IBD. And I guess what we're trying to do is disentangle that Venn diagram a little bit. And what I'm very interested in trying to understand is how different risk factors work in concert with one another to increase disease risk in individuals. And so, for example, we know the microbes are important. Without microbes, you don't get the disease. And this is true in animal models of IBD and also in humans. Get rid of the microbes and the disease goes away. So we know the microbes are involved. We know that they're involved in the inflammation. But what is it about the Crohn's disease gut that allows these particular microbes to thrive? And that's where we think the other risk factors come in. So we are very much interested in genetics, although sometimes it's very difficult to model that in animals. There's lots of caveats to that. So we have taken the approach to trying to understand the combinatorial effect, or the combination of additive risk factors in our preclinical animal models. Back when we first started this work in about 5 years after we started it, around 2012, 2013, we made the decision that if we wanted to eventually get ourselves into a position to translate some of the work we're doing in the lab to the biotech and the pharmaceutical industry, who will be necessary to take any advances to the next level, then we kind of viewed our role as providing the best quality preclinical data and evidence and information through animal models that would help de-risk some of the investment from the pharmaceutical industry in some of the things that we were trying to translate into patients eventually. And so that approach has been very productive and very successful, I think. And many of the discoveries we have made in the preclinical animal models, they do bear out when you look in trying to extrapolate them to human samples. So we think we do think the animal models are robust, and we've invested a lot of time and energy into making them better. But in those models, we are able to start to build in additive risk factors. So I'll give you just a couple examples. Antibiotic use is a risk factor for Crohn's disease. This bears out in both in the pediatric setting and also in the adult setting. And that's obviously an easy area that we can start to then use in the animal model to understand what's happening. And so we've published some work looking at what happens in the mammalian gut when it's colonized with AIEC, so these Crohn's associated bacteria, and the animal is given repeated courses of antibiotics over a period of weeks or even sometimes longer. How does that change the makeup of the microbiota? And how does it change the behavior of host immunity? And we've come to the conclusion, very interestingly, that antibiotics actually make AIEC colonization worse in the mammalian gut. And I think this is an important clinical observation as well, because these bacteria tend to be multi-drug resistant when you isolate them from Crohn's patients. By definition, they'd be superbugs. I mean, they're resistant to 4, 5, 6, sometimes more different clinical classes of antibiotics. And so obviously, if you're exposed to antibiotics and the bacteria in your gut are resistant to them, it creates an opportunity and a niche for those bacteria to expand. And that's exactly what we see in our animal models. So that's one example where we can start to look at these, again, deconstructing the Venn diagram, that allows us to look at AIEC bacteria in concert with antibiotic exposure. So that's one example. We've also looked at diet. So we've looked at AIEC infection related to diet, and we've studied high fat diet, low fiber diet, high sugar diets, and we have made interesting observations there as well, which I think have important clinical utility. We've looked at
what are some of those observations? I'm curious.
Well, so it's interesting, and there's been work published on this before, looking at the so-called Western diet, which is high fat, high sugar diets that are high in high fructose corn syrup. And in some of those studies, they have made the association or the observation that those kinds of diets also allow for bacteria like AIEC, Crohn's associated E. coli, to expand in that environment and in the face of that diet. We were also very interested in this, and what we found in our work is that, in fact, it wasn't necessarily the high fat and high sugar content of those diets. Those diets also tend to be low in fiber. And so we, in coming up with some more, I guess, deconstructed dietary interventions, we observed and we found that, in fact, it wasn't the high fat nature of the diet itself, it was the low fiber nature of the diet. And so that's, I think, an interesting observation. Foodborne pathogens. So the common foodborne pathogens that people get from, salad bars and raw hamburger and those kinds of things, raw chicken. Those types of infections, although they don't cause Crohn's disease, increase one's risk for Crohn's disease later in life. There's fascinating epidemiological studies looking at this, showing that even just one bout of food poisoning by common food poisoning bacteria like pathogenic E. coli or Salmonella enterica or Campylobacter jejuni, which you commonly find in chickens. Just one episode of food poisoning increases your risk significantly of developing Crohn's disease out to 10 years afterwards. So, from a microbiology point of view, and someone who's very interested in infectious diseases, it's fascinating to think that, an acute infection that, at most is going to make you feel unwell for about a week, 10 days tops, imprints this long-lasting disease risk for Crohn's disease out a decade from when it first happened. And so we got very interested to try and understand that in our models as well. And we've published work looking at what happens to an animal when they are colonized with Crohn's associated E. coli and then are exposed to secondary foodborne pathogenic infections. And the very fascinating thing that we found is that in fact, those types of, sometimes we call them polymicrobial infections because they're made up of they're being caused by more than one bacteria at the same time. In those polymicrobial infection settings, the foodborne pathogen actually didn't make the disease worse. It was the presence of these Crohn's associated E. coli. And all of this work has led us to understand that these Crohn's associated E. coli thrive in the inflamed gut environment. So anything in the gut that will generate inflammation, like in some cases antibiotic exposure, like foodborne pathogen infections, anything that will generate inflammation will allow these Crohn's associated bacteria to thrive. And that has then, launched us into another whole, part of the lab where we're trying to understand, because most bacteria don't do well in an inflamed environment. It's highly rich in immune cells, and immune cells are, they're programmed killers. What they do is they surveil, they keep us healthy, they keep us safe. They find a foreign microbe and they eat it or destroy it, one of the two or both. And so how is it that these types of bacteria that are associated with Crohn's disease, how is it that they thrive in an inflamed environment? They must have ways to evade host immunity so that they're not eliminated by the inflammatory immune response. And so that's another whole area where we're very, very interested in. And then finally, our most recent work, which is not yet published, but it was just accepted for publication in a prestigious journal. We are very interested to understand this link between psychological stress and IBD symptomology. And so, after having talked to a lot of gastroenterologists, one of the common things that they keep saying is, because I don't see patients, I'm not a gastroenterologist, but I have colleagues who are and do see patients. And one of the common things I hear a lot, and it's also in the literature, is that, preceding people can go into remission and be relatively well, and then will come to their gastroenterologist in relapse with acute flares. And in doing a clinical workup and history, it becomes common that people that they mention that they are experiencing episodes of psychological stress. It can be acute or it could be more prolonged due to any number of reasons. And it's not clear, how is it that the episodes of psychological stress, how is it our brain is connected to our gut, and how is it that this, bouts of psychological stress can influence disease activity in the GI tract. So this is something we have also then spent, probably 4 years now. We've been working on this in the lab pretty quietly because we think it's a pretty competitive area. And I think this first paper will be, I hope, very well received. We've made some really interesting discoveries in it, linking psychological stress to disease activity in Crohn's disease in our preclinical models. And most importantly, we've discovered a way to actually reverse the negative effects of psychological stress in those models.
So, that all sounds really good and completely fascinating. And what I'm taking away from everything you're saying is essentially anything that's going to damage the gut or cause inflammation in the gut then has the potential to allow for some pathogenic bacteria to take hold. So I have two follow-up questions from that. One is, and I've always been curious about this, and as a microbiologist, maybe you can explain it to me, and maybe I can ask a pathologist someday, but why do we not pick up something like AIEC in a standard biopsy? When patients are going in for colonoscopies, they're doing biopsies, and patients don't hear, well, we found the presence of AEIC or anything else for that matter
Mhm.
in your gut. Why is that? And do you think that we should be finding, a clinical tool or a clinical way to start testing for some of these things, even though we're at the association, not causation stage?
Sure. I think I have maybe two responses to this. And the first one is, I think in most instances, the reason that patients don't hear that they've got AIC in their biopsy sample is because it's simply not looked for. A lot of times those biopsy samples are going to a pathologist to look for disease activity and inflammation and ulceration and the things associated with a Crohn's diagnosis or clinical scoring algorithm. They don't necessarily then go to the microbiology lab for a full microbiological workup. So I guess that's one thing, is you only can find what you look for. The second challenge is that, particularly with adherent invasive E. coli, is that we don't have a genetic marker for AIEC. Mostly every other bacterial pathogen you can think of, be it Salmonella typhimurium, Salmonella typhi, Yersinia, enterohemorrhagic E. coli, I mean, you name it. They have, one or more, diagnostic genes that if that gene is present in that bacteria, you know what you're dealing with. And that's simply not the case for AIEC. So far, despite a lot of whole genome sequencing of the bacteria from many clinical studies and many clinical samples, it's been very difficult, if not impossible at this point, to define AIC at the genetic level. And that challenges our ability to do what you're suggesting and to diagnose it and to find it in biopsies. Because if it was simple as a simple PCR test, everybody knows what PCR is now because of COVID. If it was as simple as diagnosing AIC or the presence of it by a PCR test, it would be trivial. But because that's not the case, the only way to define whether or not you have AIC in a clinical sample is to first take the sample, isolate the bacteria on a plate using old modern methods of just simply plating on growth media, solid media, to grow up the bacteria on a plate. And then you take those individual colonies and subject them to a battery of tests, each one of which is technically challenging. And if they meet all of the criteria for those tests, for example, they have to be able to adhere to gut epithelial cells, they have to be able to invade and survive inside macrophages, which are a type of immune cell of the body. And then inside macrophages, they have to be able to trigger the release of TNF alpha, tumor necrosis factor alpha. And that's kind of like the three criteria that if you tick all those boxes, and you've probably got adherent invasive E. coli. And so, quite honestly, those are tricky things to do, and they're not black and white, and there's shades of gray there. And so it's tricky to do. It's just without the genetic marker or markers, it's been very difficult to do that kind of diagnosis. But that's not to say that people don't. And so in clinical studies, when people are looking for AIC in biopsies, using those things I just mentioned, they find them all the time. And so they are there. It's just not everybody looks for them.
That's great. Very, very helpful. And then I think my related question is, going back to when you hear a lot of presentations, you see that Venn diagram. Similarly, when we read papers, very often in the first paragraph, you will see that, Crohn's is of unknown etiology, but it's thought that it's a aberrant immune response to commensal bacteria. Mm-hmm. And as hard as I've tried, I've not been able to find a source for that theory, for that, which is often actually stated not as a theory, but as a fact. And I'm wondering, in view of sort of your work, is it your thinking that it is not, that it is not commensal bacteria. It's actually pathogenic bacteria. Maybe AIEC, I think I was saying it wrong earlier. AIEC is one culprit, and maybe there are others. What's your, and again, we don't know, obviously there's no definitive causation, but what's your hypothesis, I guess, around really this notion of the aberrant immune response to commensal bacteria that we hear about a lot in sort of identifying what Crohn's may be?
Yeah, I mean, it's a good point, and I think my answer is it's probably both. I don't know, that answer might not be satisfying, but hopefully I can try and unpack it a little bit. I think the key takeaway or the key message really is that in the healthy gut, we've got a single layer of epithelial cells that differentiate a line of defense between our body and the 100 trillion bacteria in the lumen of our GI tract. And it's a formidable barrier wall, and that's the wall that keeps us healthy. That's the barrier that keeps us healthy. And when you look at what that barrier does, it's not just a physical barrier where the cells are so tightly, sticking together that there's no physical space between them. The barrier has other kinds of antibiotic properties to it. It secretes lots of mucus, which creates again another physical barrier between bacteria and the epithelial surface. That mucus layer can become saturated with things like antimicrobial peptides, which are our body's natural antibiotics that we make in the gut, and they concentrate in the mucus layer. So even if bacteria can swim through the mucus, they have to confront these really potent antibiotic defenses that our bodies make. And so you can see that the body invests a lot of energy into building this wall and maintaining this barrier wall. And if you have bacteria that can break down the barrier, either by disarming the antibacterial defenses or creating breaches in the barrier between epithelial cells, which some bacteria can do, if bacteria can infect epithelial cells and damage them so that the epithelial cells then can't promote barrier function or barrier repair, then that could be a bacteria like adherent invasive E. coli. It can do all those things. It can disarm the immune system. We've demonstrated that in the lab. It can swim through the mucus layer. It can attach to the barrier and break it down. And as soon as the barrier is broken down, then you do have the opportunity. It creates an environment where, yes, your body can start to mount vigorous inflammatory responses to members of the commensal microbiota. In the gut, it's all about, like in real estate, it's all about location, location, location. And our bodies want to keep gut bacteria in the lumen of the gut. I mean, that's where they belong. That's where they're doing their job. That's where they're, providing, resistance to other infections and helping us with energy harvest from our diet and doing all the good things that they're associated with for health. Where our bodies don't want them is anywhere close to the epithelial surface, because as soon as they get close to the epithelial surface, there's the potential for damage and the potential for those bacteria then to invade deeper beyond the epithelium into spaces where they definitely shouldn't be. And again, evolution, the human evolution has provided these amazing defenses. So once bacteria do breach those barriers, they're vigorously attacked. And so I think it's both. I think if you have bacteria which can mess around with the barrier integrity in concert with bacteria that take advantage then of that barrier breach, they can't do it themselves, but once it's breached, they can move around. Then I think that's where, the contribution of the commensal microbes to disease-associated inflammation, to me, that's how I conceptualize it. I think that's how I think it all sort of fits together.
And that is very logical. It makes sense to me. Have you been able to find, or do you have a hypothesis on, you refer to it as a superbug, on what are potentially some ways, some conceptual therapeutic ideas about how we may be able to target it?
Yeah, that's a great question. So just before the pandemic put us all in our basements, I went to the Innovations in IBD meeting in New York, where it's a small meeting of about 100, 120 people, lots of company folks there talking about, next generation therapeutics, where the field is going for treatment options. And I came away from that meeting pretty clear in my mind that microbiome targeting therapeutics was kind of the next holy grail in IBD. It's where a lot of people were spending a lot of time thinking about. There were still some things looking at anti-inflammatories and new biologics that are hitting different targets. But those therapies are all still just trying to quiet down inflammation that's already there. It's not dealing with the source of the inflammation. And I think people's minds are more moving towards the microbiome as potentially being, a major source of the inflammation. And if you can figure out what, if there is a specific component of that microbiome consortium that's the problematic component, how do you get rid of it? We know antibiotics don't really work that well because they carpet bomb the entire microbiota. Even they kill the good guys and they kill the bad guys too. So that's kind of like, more broad spectrum antibiotics is not what we need. So people are talking about narrow spectrum therapeutics. A term that's been used is this idea of precision editing of the microbiota. So if you know exactly the kind of bacteria that you want to target, there's ways to then you could then conceive of ways to precisionally with precision go in and delete or edit out that problematic bacteria and see if that has beneficial effects. So that's where a lot of folks are sort of moving towards, and we are working with several big and small biotech and pharma companies who are also very interested in narrow spectrum precision therapeutics in the context of Crohn's disease. And I can't share any of that stuff with you now, but I can tell you it's very exciting.
Well, I mean, I'm certainly very excited. When it comes to the microbiome, I'm a big believer in it personally. I think there is a role for diet to modulate the microbiome. I think there's obviously would be ideal if we could have therapeutics to do that as well. I think one of the things I frankly worry about, and I'd like to hear your thoughts on the microbiome side, is that it's really it's something you could research forever. I mean, we're sort of in some ways in the relative infancy of understanding the complexity that is the microbiome as really an ecosystem.
Sure.
How does AIEC interact with other bacteria and fungi and viruses? And, maybe there are maybe it's more of them working in concert together, or, we hear a lot about biofilms, and maybe there's a role for the whole microenvironment in a biofilm that could be problematic. How do we get to a point? And obviously, as a parent, we have a sense of urgency around these things. How do we move from interesting kind of theoretical research to that again could last decades, to really trying to get to understanding whether there is a role either in causation or in exacerbation of disease? And if the answer to either one of those is yes, how do we really move quickly to trying to find a target? And I know those are impossible questions to answer.
Yeah, I mean, those questions will take a career to answer in the lab. But you're right. I mean, there is a sense of urgency, and I guess I'm continually buoyed by the sheer volume and number of really smart folks who are studying these problems in the lab. Obviously, this is not something any one person can take on themselves and expect to solve all the issues. But as a community of researchers, I think we can definitely make a huge dent. And also for your listeners too. I mean, it probably is not inherently obvious. The IBD research community, in my experience, is highly collaborative, and it's not true. I mean, I've got the other half of my lab doesn't work on IBD. We work on host pathogen interactions and other kinds of superbugs. And can't say it's exactly the same kind of collaborative spirit, but I mean, it's okay, but it's not. The IBD community seems to always sort of rally around this shared vision and the shared purpose of we need to find a cure. I mean, that's it. We need to find a cure, and this disease won't go away without it, and it's only going to get worse, and prevalence will only continue to escalate. It's compounding prevalence. It's once you have it for life. So there is a sense of urgency. I mean, I know not everyone likes preclinical animal models, but I guess going back to your question specifically, I am a believer in them. I think they have their caveats. They're a model, of course, but essentially so is everything else. And I think deconstructing the system and allowing you to control certain variables in a sort of defined preclinical animal setting, it can go a long way to helping to understand and unpack the what's happening in humans for the translational arm. The other important thing about I think about preclinical animal models, and certainly this has been my experience, is when done well, and if extrapolated to the extent possible to humans, it does help de-risk partnerships and investment from the private sector. And the investment from the private sector can't really be overstated. I mean, it's so important. We've done lots of sponsored research agreements with many companies over the years. Investigator-driven funding is harder and harder to come by. I get grants from Canadian Institutes of Health Research, which is sort of your equivalent of the NIH in the States. Crohn's and Colitis Canada also generously funds our research, but these grants are not easy to come by. They're certainly not slam dunks when they get submitted. And in any one cycle of CIHR funding in a year, it's possible that not one IBD related grant could get funded just on the way things shake down and the scores shake down. So that's a problem, right? I mean, if we need sort of constant interest and constant investment in the research engine so that we don't stall out. And I think that's where there is a role for the private sector. And if they can see some promising preclinical data that will, or a model, for example. I mean, much of our collaboration has come from because we have developed these unique animal models in the preclinical setting that have allowed companies to test some of their pipeline molecules in. And it's kind of like the only way that it's going to advance to the next stage, which would be clinical trials in humans. So I do think there's a role for it. I'm fully aware of the caveats to them, and but I do think that there's a role for them, and I think they can really help advance the translational aspects too.
Yeah, that makes a lot of sense. And maybe I'm jumping back a little bit, but have you been able to demonstrate in an animal model that AIEC causes Crohn's at least, or Crohn's-like colitis, or however you would term it in an animal model?
Yes, we have. That was the first paper we published on this in 2013, where we, and again, we knew this was probably going to be controversial. We knew it was going to be tough. So the postdoc who decided to take on this project, the very talented and brave postdoc in my lab, Dr. Sherry Small, we set out to develop this animal model, and not just do it in one mouse line, but in the paper we actually described the model in 5 different conventional mouse lines, not just one, and tried our best to be as comprehensive as possible in scoring and defining inflammation in all regions of the GI tract in the model. So we looked at both the ileum and the colon and the cecum. And so yes, we have, and it generates inflammation. It causes fibrosis as well over time, which is not something that is a lot of infectious animal models don't lead to fibrosis in the gut, but AIC does. So we demonstrated that in the paper as well, which is, I think, a very important step to link it to Crohn's disease, because fibrosis is uniquely associated with Crohn's. So yeah, I think there is good evidence that it's doing something not healthy for our GI tracts.
Yeah, and that's a great additional piece of information. And I've read that paper. It's a great paper. It's one of the ways that that we know you, that we know of you. And the fibrosis point is really important and interesting because, of course, that's one of the things that is really not well treated in IBD. Mm-hmm. There's really no good therapeutic for stricturing Crohn's. And so perhaps, is it your thought that this could be an avenue that could potentially down the road address that as well? So if you address the AIEC, you potentially can address or at least avoid the stricturing Crohn's.
Yeah, I mean, it's one of the areas we've been thinking a lot about in the lab is, if you get rid of, how can you, if you need to modulate, not only the bacteria, but also the things that go along with the disease, like stricturing and fibrosis and the inflammatory response, if you modulate one, do you solve the other? And so if we get rid of AIC, does the inflammation go away? If we dampen the inflammation, does AIC actually go away? Because we know AIC thrives in inflammation. And so we are looking at these, it's almost like, in a way, it's kind of like thinking about it like in three-dimensional chess, because you pull on one thing and something else is going to happen. So you have to perturb the system in a way. And then when you perturb the system, many things change. And so we are very interested to know, if we pull on one thing, do we affect the other? And I do think it's potentially a good model for folks that are, companies that have a pipeline in anti-fibrotic drugs. I think it could be useful. But as you say, it's a really tricky thing to try and reverse once fibrosis starts. It's really hard to reverse. Surgery is kind of the only thing that we've got right now. And then it always comes back. So I know there are companies working really hard on fibrosis.
Yes, yeah, there are a couple companies and researchers in academia looking at it, but that's really helpful. I think you've certainly answered a lot of my questions. I won't keep you for too long, but is there anything else that you would like people in the IBD community to know about you, about your lab, about some of your collaborators, where they can find you, and any other information that you want to pass along? Please feel free.
Well, certainly people can, and if they Google that name, they'll find me pretty quick. I'm a professor of biochemistry and biomedical sciences at McMaster University in Hamilton, Ontario, Canada. I've been here for 16 years. I what else do I want people to know? I mean, I guess I want patients and people to know that there's a lot of people who are working really hard on solving this disease, and we won't stop. We won't stop. And not only am I very passionate about this, but everybody who's working on this stuff in the lab is also equally passionate about it. And there's a lot of smart people working on this problem, and we will solve it.
Thank you. That's a good place to end.
Yes. Thank you, Brian, so much for all of this. I found it really fascinating. It was excellent. And it does give me hope just to know that I really like I really liked hearing the fact that the researchers in the IBD community, in the IBD sphere, that they collaborate, because I think that's the only way we're going to get to the cure, because it's just too complicated otherwise.
I agree.
And one thing I wanted to ask is, do these cells go dormant? Is that a possibility? And do they hide in macrophages?
It's a good question. I don't really know the answer yet. I don't really understand yet where they necessarily are hiding out in the body or where they go. I know a lot about what kind of cells they can infect, but I don't necessarily know exactly, where they are and how metabolically active they are once they colonize the GI tract. It's actually a very, your question kind of is like at the limit of knowledge right now. So that's great. I just got a grant from Crohn's and Colitis Canada, and I have a grant submitted to the Canadian Institutes of Health Research to answer this very question. Is where are these cells living in the GI tract, and what kind of thing, are they metabolically active? And if so, what are they, what kind of metabolism are they doing, with the eventual, goal that if we understand how it is they're evading our body's natural defenses, that gives us a place to look to try and combat them.
Is it possible that they pervert the CD8 cells, T cells, or something like that? They do some sort of dysregulate them in some way, do you think? Or is that
I think anything's possible. I've been studying host pathogen interactions for the better part of 25 years. And, for every host immune defense that you can think of that, that have evolved in us over millennia to keep us healthy, bacterial pathogens have evolved ways to overcome them.
Mhm.
They are, in a way, bacteria are the ultimate little cell biologists themselves. They understand our immune system better than we do in some cases, and they have evolved ways to overcome our body's natural defenses. So I would expect that, if those kinds of, if a particular immune cell is important for host defense, bacteria that have, long-term associations with hosts over chronic timescales have figured out ways to get around it. And so our job is to figure out how they're doing it and reverse it again.
Or learn to live with them.
Maybe.
Right? Is there one school of thought that you just adapt and learn to live with them, meaning you find a way to not have the immune response, kind of get out of control in response to trying to just, chronically rid yourself of these pathogens?
Yeah, that's kind of an interesting topic about, the evolution of bacterial pathogenesis. And that's, a lot of people have spent a lot of time thinking about that too. And a lot of it depends on how bacteria and viruses are transmitted from person to person. A lot of that will dictate sort of the degree to which the bacteria evolved to be more benign versus more aggressive in their behavior. So
how would you, because Crohn's is spreading worldwide, it wasn't particularly prevalent in Asia in the past, but now it's becoming more prevalent, much more prevalent. Do you have any thoughts on that as to why that's happening?
Obviously there's no definitive answers. I suspect that a lot of it has to do with, again, probably diet to some extent. I think it probably has a lot to do with access to some medicines that become more common and prevalent in industrialized societies, like antibiotic use, for example. I don't know the answer, but I do. But from a research point of view, it's kind of an interesting cohort to try and study and understand, because the disease, I would say, is, it's well established in developed countries, obviously. In other countries right now, like as you mentioned, Asia and also South America, I would say it's emerging. And to try and study this in an emergent population, I think allows us to ask different kinds of questions and maybe get different kinds of answers as to what's happening. So I don't know the answer to your question specifically, but I do think that the emergence of this disease in certain geographical areas now can inspire different questions in research to help us better understand the disease.
Brian, I have a question for you. What is the next big thing that you hope to really figure out about this pathogen? Like, what do you think is like the next key that will unlock the next door for you?
Yeah, it's a good question. I'm really interested in sort of the communication between bacteria and the host. So, when bacteria land in our GI tract, they immediately start sensing where they are. They have lots of sensors on their body, on their cell surfaces, and they immediately start to, figure out where they are based on the chemical cues that they sense, based on, all kinds of things, oxygen tension, acid, pH, and things like that. So I'm really interested to try and understand, like, what is it about AIC that make them unique among E. coli that allow them to thrive in the inflamed gut? It's got to be something that, it has to be a genetic program that they launch once they get inside our GI tract that then program them to evade our host defenses. And I think there's probably ways to, if we understand what those host defense, those ways that they overcome our host defenses, I do believe that there's ways that we can then target those systems in the bacteria to disarm their evasion strategies as an alternative to antibiotics, because we know antibiotics are going to be tricky in this particular case.
Do you think that there's a lot of interest in phage therapy right now? Do you think that phage therapy could have some utility here?
Yes, there are folks even at McMaster who are very interested in phage therapy, who are studying phage, targeting very, and that's the cool thing about phage is by, the way that they work, they're narrow spectrum, like they're very, like they're laser beam narrow spectrum. So, they tend to infect and work on a very limited, range of bacteria, and in some cases, not just species specific, but in some cases, strain specific. So within one species, just different strains can be more or less susceptible. So I do think, if you're going after a narrow spectrum kind of therapeutic, I do think phage probably are something to think about, and lots of people are working on this.
Is this particular, I always mix it up to AIEC,
Yeah.
is this particular pathogen something, and I might have missed this earlier, is this something that is resident in everyone's gut, or is this something that we pick up in some way, or is there any way to determine that?
It's not resident in everyone, although, in studies where people have looked for it in Crohn's patients, the control arm tends to be healthy individuals that are undergoing, not everyone's going to raise their hand and say, yes, please give me a colonoscopy. But if you're undergoing, a diagnostic colonoscopy for, say, colon cancer screening, then oftentimes those individuals are used as controls in those types of studies. So they don't have Crohn's disease, but they're getting scoped for different reasons. And in about 6% of those biopsy samples from individuals, you can find AIC. So it's not like it's missing from healthy individuals. It's there, but it's just really low abundance or low frequency, or not easy to find. So, that's one thing. And then the second part of your question was, remind me?
Do you is this something that you pick up from the environment?
Oh, yeah.
Somewhere?
So that's another fascinating question, and the answer is we have no idea. I used to work in public health, so like I'm constantly interested in like disease provenance and bacterial provenance. Like, where do these things come from? How do we get them? There's been a lot of good work trying to figure this out, and I would say, answers are not firm, but there's been a really, there's a researcher at Cornell, Dr. Ken Simpson, who's been working on this question. He's found AIC bacteria associated with companion animals, mainly dogs. So you can find AIC in pets. So again, that's, that could be a potential route to transmission to humans. We don't know. I'm not aware of any definitive transmission event from animal to human, but the fact that you can find AIC in companion animals, I think, is kind of interesting. So it could have something to do with that.
That's really interesting. And what would you say is like the biggest need of your lab right now?
Funding. What keeps us going, right? Research is incredibly expensive, and we are constantly hustling. constantly hustling for research funding. So yeah, funding. And I'm always on the lookout for talented people who want to contribute to this kind of research. It's one of the things we never stop doing. We never stop writing grants, and we never stop looking for talented people who want to do training and make a dent in this problem.
That's wonderful. Thank you.
Agreed. I mean, your collaboration comment earlier, I think is so critical, because there are so many, I joke, and it's completely a joke, but that when my daughter was diagnosed, I became a nutritionist, microbiologist, geneticist, immunologist, and gastroenterologist to try to help her. And of course, I didn't do any of those things. I'm not an expert in any of those things. But I think what it does sort of show me is that there are a lot of silos out there. There are a lot of people doing phenomenally good immunology research, like what we funded at Stanford. There are people like you doing incredibly great microbiology research, et cetera. Genetics, which you talked about earlier. And at some point, these Venn diagrams do have to kind of come together. It's a puzzle. We always talk about it as a puzzle. We have to fit the pieces of the puzzle together.
Yeah.
And I think, one of the things we talk about a lot is how to get people together to try to start putting those pieces of the puzzle together, so that everyone isn't just off kind of working on one little isolated piece of the puzzle. Although that's too. I mean, you have to get the puzzle pieces before you can put them, into the bigger picture. But it's time to kind of start assembling the puzzle.
I totally agree with that. And I've always been a firm believer that big breakthroughs happen at the boundaries and interfaces of two disciplines or more than one discipline. And it's, that's where I do think that's where the big discoveries happen. And we can do that at a micro level within the lab too. I mean, I've always tried to have, I'm a microbiologist and a biochemist, but my lab's always had, immunologists, biochemists, microbiologists, cell biologists, structural biologists, all at once. And so, the way I look at it is, if I just hired microbiologists, the only thing we would think about all the time is microbiology. We would attack problems only from one direction, only because that's the way we're trained to think. And so, with a diverse group, we can think of ways of tackling problems from different angles, and push the needle faster, more efficiently, different ways, thinking differently about what's been done before. I mean, that's one of the things I always loved about science and research is we're obviously there for to do science, but it's a highly creative endeavor too. I mean, we have to think about problems in different ways, because if you just think about them the way it's always been thought of, you won't solve the problem, you won't push the needle. So we do have to think about things differently, and get creative, pull in new technology as it gets developed from other disciplines and apply it to our problems. And I think that's how we make advances.
Couldn't agree more.
Oh, well said.
So well said. We'll probably have to make sure we edit this all together and put that last comment in there too, because I couldn't agree more. I'm conscious of your time. I know we've taken up a lot of it already, but I'm so appreciative. First of all, we could talk about this all day because it's so fascinating. I mean, it's just intellectually fascinating, other than obviously being important to Crohn's patients and parents. But thank you so much for the work that you do, and for being creative and collaborating and asking tough questions, even, pushing the envelope if you have to. I think, that's the way progress is made. So thank you again.